Hello everyone, I am Jay and I am working in Professor Mang's lab at IBC Academia Sinica. We are working on tyrosine phosphatase enzyme called TCP/TP and trying to understand how its activity is regulated so that we can tune it for the cancer treatment. Actually, this protein is known to dephosphorylate multiple substrates associated with cancer like various RTK and other tyrosine kinase and other. But unfortunately, when this protein is expressed, it remains in inactive state. Therefore, it cannot do its function. But it has been known that if it interacts with integrin alpha 1, it can get activated and it can dephosphorylate its substrates such as RTK efficiently. However, why this protein is in inactive state when expressed and how it is getting is activated is unknown, and that's what we are trying to address by this study. Coming to our first question, that why this protein remain in autoinhibitory state when it is expressed, we found that the C-terminal tail of this protein interact with catalytic domain and form autoinhibitory state, autoinhibitory conformation, due to which it auto-inhibits enzymatic activity, and that's why you can see that catalytic domain lacking C-terminal tail has much higher activity compared to the full length. To find out where exactly this C-terminal tail interact on catalytic domain to auto-inhibit its enzymatic activity, we perform an NMR experiment and found that it bound two distinct surfaces, one N surface near to the active site, one B surface behind the active site. Based on our data, we have developed a model to explain molecular mechanism of TCP/TP autoinhibition. Our model shows that C-terminal tail of TCP/TP is flexible in nature and it moves around the active site just like the windshield wiper of the car and prevent the access of substrate. As a result, the enzymatic activity of this protein gets auto-inhibited. Coming to our second question, that how TCP/TP get activated by interacting with integrin alpha 1, we examine the interaction of integrin alpha 1 cytoplasmic tail with TCP/TP catalytic domain and found that it binds to the same site on the catalytic domain where the C-terminal tail was binding, suggesting that interaction of integrin alpha 1 cytoplasmic tail to the catalytic domain will disrupt the intramolecular autoinhibitory conformation, causing the enzyme to be activated. Our study concludes that inactive state of TCP/TP is formed due to intramolecular interaction between C-terminal tail and catalytic domain, but when integrin alpha 1 interact with it, it disrupts the intramolecular interaction and make this enzyme fully active for efficient dephosphorylation of substrate. Thank you for watching.